All right, so we are going to go back to cell division in this particular unit, and we're going to specifically cover meiosis. So a couple things before we start this. The first is that we've covered the structure of DNA. We talked about how DNA had to replicate prior to the cell dividing for the mitosis cell process. The same thing has to happen here, okay? Then after we covered that cell division unit, then we went from DNA to RNA to proteins with our protein synthesis unit. And now we're going back to needing to know the information that we covered at the very beginning of January. We're still going to pull in some protein synthesis and some things like that and just continue to make connections. But now we're going back to cell division. All right, so that's why it has this statement up here in pink. All right, so meiosis, by definition, it's the process of reduction division in which the number of chromosomes is going to be cut in half through the separation of homologous chromosomes as the cell divides, right? So that's a long definition, okay? So the reduction part is that the number of chromosomes is reduced by half. And then the division part of that, in quotes, is that the cell is dividing, right? So there are two stages of meiosis, meiosis 1 and meiosis 2, and there are two separate rounds of division. Right, so meiosis 1 is going to start with one single diploid cell. Remember, we already covered what diploid meant in the first set of notes from this semester. So the N, as a reminder, is the number of different types of chromosomes in a given diploid cell. Okay, so if we look at this diagram, there's three different types: pink, purple, and yellow. There's two of each type, so 2 times 3 gives us a diploid number of 6 right here, right? And so in our bodies, in males, the testicles are going to be diploid or filled with diploid cells or made of diploid cells, and then ovaries in females are made of diploid cells. And from those diploid germ cells, haploid cells are going to be formed via meiosis, okay? So at the end of meiosis 2, there's now going to be 4 haploid cells, where we started with 1 diploid cell at the beginning of meiosis 1. Okay, so haploid cells are just N, or 1N. Right, the only 2 haploid cells in the human body are eggs in females or sperm in males. Right, so here's a haploid cell with a haploid number of 6, 1 of each type, 1 pink, 1 purple, and 1 yellow. All right, so then if you have 2 haploid cells, an egg cell and a sperm cell, that each have a haploid number of 6, and those fuse together, then when that zygote forms, that zygote's going to be diploid, and then that cell would grow and divide by mitosis. All right, and that's what we modeled on the whiteboards. That's what you drew on your diagram with the diploid number of 8, okay? So go back to those notes about diploid, all right? Homologous chromosomes are present in diploid cells. All right, so then haploid cells, by definition, are going to be present when I can't get this in the right place, hang on, when there's only a single copy of each type of chromosome, okay? So again, N, right, or 1N. So in humans, the egg and sperm cells are going to have 23 chromosomes. Right, so when a sperm cell from a male and an egg cell from a female, each with 23 chromosomes, when those fuse together to make the zygote, and I know you can't see that very well, okay, we're going to have 23 plus 23, that's going to then equal 46 total chromosomes in the diploid cell. Okay, so egg cell plus sperm cell, the male's on this side. Sperm cell plus egg cell in that diagram, right? 23 plus 23 gives you a diploid zygote with 46. It'll grow and divide by mitosis to become a multicellular human in the case of this context. Of course, this is going to happen in any other type of organism as well that's multicellular. To get from a zygote to that multicellular organism, it's going to have to grow and divide by mitosis. All right, and so all of that we've already covered. And so we're going to go back to cell division, yet we are going to cover meiosis, okay? I don't know why I said go back to cell division, sorry. So we're going to start going back to interphase is what I was meaning to say, okay? So you have short and sweet statement here. interphase prior to meiosis taking place is going to just happen in the same way that it happens prior to mitosis taking place. DNA is going to have to replicate, the cell's going to have to get ready to divide, so go back to those notes, right? In meiosis, again, we're going to have meiosis 1, and then within meiosis 1, we're going to have prophase 1 through telophase 1, and then we're going to have meiosis 2, okay, and everything's going to be followed with a 2. Right, if you leave out the 1s and 2s, then everything, your answer is automatically wrong, because there's no such thing as just prophase, for example, in meiosis. So in prophase 1, the chromosomes, as we talked about in prophase of mitosis, they're going to coil up and become really dense, like what we see over here. Spindle fibers are going to form, centrioles are going to migrate to the opposite poles of the cell, right? And so then each homologous pair of chromosomes is going to form what's called a tetrad. And a tetrad is made of 2 homologous chromosomes, each made of 2 sister chromatids. So we see 2 tetrads here. We see 1 tetrad down here, right? So the prefix tetra means 4, right? And if you've ever played the game Tetris, then every shape in Tetris is made of 4 squares. Right, there's a straight line, there's one that looks kind of like the windows in my room, a big square with 4 squares within it. There's a little S and a little Z, just depending on how you look at those. There's a T, then there's L's that face in 2 different directions, okay? So that's what that prefix means, right? So what's there 4 of? So there are 4 sister chromatids in a tetrad, okay? So in this picture down here, ignore what crossing over is at this point. That's 1 tetrad on the left, another tetrad, a 3rd tetrad, and a 4th tetrad, okay? So that's where the tetra comes from, okay? So that's a homologous pair of replicated chromosomes, right? So in a tetrad, the sister chromatids and or non-sister chromatids in 2 different chromosomes are going to pair really tightly. And when that happens, they may exchange genetic information through a process called crossing over, which we see here, right? And then this is the outcome of that crossing over, where now you have lowercase gray C, D, and E section on this chromosome on the left, and then the uppercase C, D, and E white section on that chromosome. So this is going to enhance genetic variation. Right, it's new combinations of alleles, right? And we're going to talk about that term soon. Basically, alleles are forms of a gene on the chromosome. Okay, so in this picture, these capital letters, that's representing dominant alleles. We're not to that yet. And then the lowercase letters are representing recessive alleles. Okay, now this is just showing this section having crossed over, but crossing over may happen on, say, just the E's, or it may be A through E. Okay, it's going to vary for every round of meiosis. Okay, and so this homologous pair is crossed over, and so is this one in this particular diagram. So let's go to metaphase 1. Okay, so in metaphase 1, now the tetrads are going to line up in the center of the cell with 1 member of each homologous pair on either side of the center. And there's 2 ways for a pair of anything to line up, right? So imagine sitting in my classroom. If everyone were in class, there would be 2 people at each table, okay? So if you sit on the left side of the table when you're facing the board, right, then somebody else would sit on the right side of the table. And the only way you 2 individuals can line up is to line up the way you're seated or to swap places, okay? And that's the same thing for a pair of anything. Like, you can put your shoes on the correct feet or you can put them on the wrong feet, all right? And so every time the homologous pairs line up in metaphase 1, each pair can line up in one of 2 ways, so that's going to enhance genetic variation. So if we think about humans, for example, with 23 pairs of chromosomes, and that there's one of 2 ways that those chromosomes can line up in this particular phase, metaphase 1, 23 different pairs, that's a lot of variation. Plus we add into that crossing over may or may not occur. Okay, so tons of ways to create new genetic combinations in the eggs or sperm, respectively. So then in anaphase 1, the homologous chromosomes are going to separate and move to opposite poles of the cell like we see here. This is where we're going to have the assurance that each new cell is going to receive only 1 chromosome from each homologous pair. Okay, so we are already working toward haploid cells with that phase, with anaphase 1. All right, so I just combined telophase 1 and cytokinesis, and I don't have a diagram on this particular slide. But then when the chromosomes reach the poles, that's going to be when telophase 1 is going to begin. The spindle fibers are going to break down. The cytoplasm is going to divide during cytokinesis, essentially at the same time as telophase 1 taking place, to make 2 haploid cells. And so by the end of telophase 1 and cytokinesis combined, we've got 2 haploid cells, each with half of the genetic information of the original pair, of the original cell, because now you have only 1 chromosome from each homologous pair present. And that's what we're seeing here in this particular diagram, okay? So now this is the end of telophase, or towards the end, the nuclear envelope's reforming. We've got the red chromosome in the top cell, or what's going to become the top cell, and the orange one and the bottom one, those were a homologous pair. And then remember, blue and green were a homologous pair. So now those are split apart. So now we've got 2 homologous cells that are forming. Once the cytokinesis event is finished, then we'll truly have 2 haploid cells. Then meiosis 2 takes place in order to get rid of the replicated nature of those chromosomes. So let's go back up here, okay, and then we'll go back to that diagram. So then prophase 2 is going to happen. Spindle fibers are going to form in each new cell. Metaphase 2, the chromosomes are going to line up in the middle of the cell. Kind of envision mitosis with that, and metaphase of mitosis. In anaphase 2, the sister chromatids are going to separate, become individual daughter chromosomes to move to opposite poles of the cell. Envision anaphase of mitosis. Okay, that's what that's going to look a little more like. And then telophase 2 in cytokinesis, the nuclear envelope is going to reform, spindle fibers are going to break down, chromosomes going to uncoil, and then the cytoplasm is going to divide again as well. And then we're going to have 1 member of each homologous pair, okay, and no longer the replicated nature of chromosomes. So let's go real quickly to this particular diagram. So here, okay, interphase or diploid numbers 4. Each of those strands of chromatin replicates, the 2 identical copies join together, and then tetrads are going to form in prophase 1, okay, like we see there. So we've got 2 tetrads. Those tetrads line up in the center of the cell. In metaphase 1, they can line up in one of 2 ways, okay, for each of those pairs, right? Then those homologous pairs are going to separate. By the end of telophase 1, we have 2 haploid nuclei, and once cytokinesis is finished, then we've got 2 haploid cells. So that when we go into prophase 2, in this next diagram, right, when we go into prophase 2, okay, that's what we're seeing here on the far left. We've got 2 haploid cells, each with only 1 member of the homologous pair, okay? Then those chromosomes line up in the center, okay, because now the replicated nature of the chromosomes needs to disappear. Sister chromatids separate in anaphase 1, and these are now, anaphase 2. Now these are individual daughter chromosomes, 1 set moving to each pole in each of these 2 cells. So then by the end of telophase 2, each cell has half of the original genetic information of the parent cell, okay? So every round of meiosis makes 4 cells. Right? In males, 4 sperm cells are going to be formed for every single round of this process. In females, only 1 egg cell is formed. The other 3 cells are really tiny, and they are called polar bodies. So that egg cell is huge. So in females, any 4 of these, if this was egg production, any 4 of these could become the egg. So let's just pretend that this one becomes the egg. It would be much larger in a diagram, and the other 3 would be really small. And then the ovaries resorb those polar bodies. They just go back into the tissue. The egg cell then is either going to be used for fertilization or it's not. Those 3 polar bodies basically are never going to be used for fertilization, okay? Same thing with sperm cells. They're either going to be used for fertilization, right, where an egg and a sperm cell fuse, or they're not, okay? So when we look down here on this particular slide at the very end, and then we'll go back to the middle part. So meiosis is not a cycle, okay? It's not in a circle on the diagram. You've just got those 4 cells on the far right side of the diagram. because they are either going to be used for reproduction or they are not. And if they're not, then they're going to die, okay? If they're used for reproduction, then again, let's think humans in this case. 23 chromosomes in an egg cell joins with 23 chromosomes in a sperm cell. 23 plus 23, you get 46 chromosomes in that zygote that can grow and divide by mitosis to turn into a human. And this is how we all got here, okay? So let's go back and kind of talk about this little section here. So meiosis and sexual reproduction, that's what allows for genetic variation in nature in multicellular organisms. So it says the total number of different genetic factors that make up a species. All right, so when we look at any population of organisms, if it is a sexually reproducing species, meaning that it's something that's multicellular, okay, and can produce gametes, and you have two parents that then form offspring, there's genetic variation out there. In us, we see that really easily in our physical characteristics, but in things like squirrels, like all gray squirrels kind of look like a squirrel to you, and you don't see that variation. And all oak trees that might be in your backyard, you don't see that variation, but it is there, right? And so then what we see in nature is that genetic variation is going to enhance the chance of survival of multicellular species. And it's going to drive the evolutionary process. So when we get through everything that we've got to do with genetics and for the next few weeks, then we're going to go to natural selection, and we're going to talk about the process of how that occurs in nature. Okay, and so we'll pull back to this at that time. If you've got any questions about, oh, I've got one more slide. Hang on. All right, so I'm not going to have you draw this particular diagram, but this is drawn like your mitosis with a diploid number of 8 on the legal sheet was. So notice we've got interphase before replication. Now we've got a red homologous pair, a green homologous pair, and a blue homologous pair, where let's say that in the mother, okay, that one of these particular chromosomes came from her mother, and then the other one came from her father to make that homologous pair. Okay, so diploid number of 6. Each of those is going to replicate, so we still have a diploid number of 6 here. Okay, we've just got identical copies of chroma 10. Okay, remember it's chroma 10 in interphase. Then we get to here, we've got our tetrads, right? Those tetrads are going to, there's no crossing over shown in this particular drawing. Tetrads are going to line up in the center of the cell in metaphase 1. Okay, and each pair again can line up in one of two ways. They're going to split apart in anaphase 1. So that then by the end of telophase 1, and then once cytokinesis is over, you've got 2 haploid cells, each with only 1 chromosome from each homologous pair, but they're still replicated. So then meiosis 2 is going to begin, and that's going to get rid of the replicated chromosomes, right? And so then just kind of go into here, those replicated chromosomes are going to line up in the center of the cell in metaphase 2, split apart in anaphase 2, so that those sister chromatids are now called individual daughter chromosomes. And then we get 4 different cells that are either going to be eggs or sperm respectively, depending on if we're talking about males or females. Okay. Notice though they all look the same here, just to make this drawing simple, but crossing over might have occurred. And there's always going to be some genetic variation that we're not going to see really well in a diagram such as this. Okay. And then you've got some notes here. Okay. There's 3 types of chromosomes present, 3 homologous pairs present, 2 of each type of chromosome in the diploid cells, and then once we get to the haploid cells, you've got 1 of each type. Okay, if you have any questions, let me know.